Guys, welcome back to another video on Arshbionyms. So in today's video, we are going to discuss about the second method to identify bacterial motility. And before I start with the video, I just want to tell that all you guys, all your words are being noted. Now that the students have been texting me on my Instagram, and also that students are commenting on my post asking me to teach more in English because few students are not able to understand the Hindi thing, okay, Hindi language. So I will make sure about it. I, being a teacher, I need to make sure that each and every student of mine, whoever is watching the video, should understand whatever I'm teaching in very easy possible way. So your words are noted. I'll make note about it. But if in case in mid of the video, whenever it happens that sometimes whenever I'm in like too deep into the topic, that Hindi thing comes out. I start speaking in Hindi. So even if in case I speak in Hindi, I will make sure that I translate the same thing in English as well. Okay. So from now onwards, that wouldn't be an issue. So anyhow guys, let us start with the video. So in the earlier video, we have discussed about the first method to identify the bacterial motility, that is hanging drop test. है ना? Now in today's video, we are going to understand Flagellar staining method. Now under the Flagellar staining method, we are going to study two different types of method. Now the first method is wet mount method and the second method is Leibson staining method. Now both the methods are like almost similar. It's just that the type of stains we use in both the methods are different. Otherwise the steps are like almost similar, not completely though, but yeah, you just need to understand the important points. And here you go with wet mount and Leibson. It's almost the same thing only. Okay, so now before we start with wet mount method, let us understand the basic structure of a flagella. Okay, now flagella here is very thin. It is thread-like, and these flagella is appendage of plasma membrane. Plasma membrane, nothing but the cell membrane, the same thing. Okay. Now the concept behind this is because flagella is too thin to be noticed through the microscope, we have to first convert the thin structure into the thick structure. So whatever the stains we are using here in wet mount method and Leibson staining method, the stains what they are doing to the flagella is they are converting this thin flagella to thick flagella. I repeat the statement again. Whatever the stains we are using in wet mount and Leibson staining method, they are converting thin flagella to thick flagella such that it would be easy for scientists to observe, or easy for all of us to observe through the microscope. That's the whole concept behind using this wet mount and Leibson staining method. Okay, so first let us start with the wet mount method and understand what exactly it is. Now wet mount method is otherwise also called as Ryu method. Okay, now in this method, we have to make a Ryu solution. Now basically that solution is made up of two different things. Ryu stain, we generally call it as Ryu stain. So Ryu stain, whenever we are making it, that is nothing but solution 1 and solution 2. So here wet mount method is otherwise also called as Ryu method. Why Ryu method? Because we are using Ryu stain. Ryu stain includes both solution 1 and solution 2. Now solution 1 includes phenol, tannic acid, aluminium potassium sulfate solution. Simple. You have to remember this. This is important. Solution 1 includes phenol, tannic acid, aluminium phosphate sulfate solution, and solution 2 includes crystal violet and ethanol. So we are going to use these two solutions to make complete Ryu stain. Now what we are doing in this method is, first I'm going to take a very clean slide and then I'm going to add a drop of water on it. ठीक है, ये मैंने add कर दिया water drop. Okay, now on the drop of water I'm going to add inoculum. Okay, I'm just going to mix the inoculum in that drop of water. So the second step would be adding the inoculum on the drop of water, and I'm going to mix it completely and spread on the slide, clean slide. Okay, now leave the glass slide undisturbed. We should not move again here and there because we just want the slide to be undisturbed so that we get to see the bacterial motility very clearly. Then after leaving the slide undisturbed, we are going to cover that particular drop or the area on the slide where we spread the inoculum through a cover slip. Now what are we going to do here is we are going to observe it under the microscope. Okay, now once after placing the cover slip and when you're seeing through the microscope, when you observe any kind of movement, when you see that the cells are moving, now when you confirm it that motility is to be seen in that particular inoculum what you kept on the slide, then you will go ahead and add Ryu stain. Are you understanding what I'm telling? आप पहले क्या करोगे? First what will you do? That you will observe it under the microscope, and when you notice that the bacterial cells are moving, then only you will add Ryu stain on top of it. Okay? Now how are we going to add Ryu stain? So basically whenever we add Ryu stain, we are not going to take out the cover slip here in this case. Just on the border of the cover slip, this is how you are going to add the Ryu stain. So because of the capillary action, action, the Ryu stain is going to move inside the cover slip. Simple as that. तो आप ना cover slip के side से Ryu stain को डाल दोगे तो Ryu stain capillary action की वजह से वो अंदर की तरफ खींचा चला जाएगा. So Ryu stain is moving inside the cover slip due to capillarity or capillary action. Simple है ना. Now what are we going to do? After you add the Ryu stain, you are going to observe it again under the microscope. Now you get to notice that the flagella appears in purple color. So basically the complete structure here will appear in purple color. So this is what is Ryu method or wet mount method. Let me show you the diagram how it is seen under the microscope so that you will have a more clarity over it. Let me just show you. So this is how the wet mount method. This is how the bacteria would look like in the wet mount method. So you see it is completely appearing. The whole flagella is appearing in purple color. ठीक है? Clear है? I hope it is clear till here. ठीक है? Now let's move ahead with the Leifson stain. It's again the same thing, only just the type of stain what you're using is little different here in Leifson stain. Now in Leifson stain, in Leifson staining method, we are using Leifson stain and counter stain. Now the Leifson stain is made up of tannic acid. Same thing, we have used tannic acid in wet mount as well, but here we are going to use basic fustian, alcohol, potassium alum. Again the same thing, potassium alum solution, basic fustian we are using alcohol and tannic acid. Now in the counter stain we are going to use methylene blue. ठीक है, सही है. Now what are we going to do is we are going to take a slide, a clean slide. We are going to keep the slide in slant position. We are not going to keep the slide horizontally on the table, but we are going to hold it in the slant position. This is how the angle of the slide should look like. Okay. Now in step 2, we are going to add inoculum on the slide. So you see here, I have added the inoculum. Now just because we are keeping the slide in a slant position, the inoculum is going to flow through the slide in this angle. So this is how the inoculum is falling from the other side because we kept the slide in the slant position. Simple as that. ठीक है? Now we are going to air dry it. Completely air dry करना है such that all the liquidy part will get dried and only the inoculum, basically the bacterial culture is there on particular slide. So you see this is how the bacterial culture would look like in the slide. This particular diagram I'm talking about. So dried slide, after the slide is getting dried, only the inoculum would look like this. Now on the dried slide, we are going to add up the Leifson stain. Now if you notice, so because we kept the slide in slant position, when we add Leifson stain like this, of course the Leifson stain will also fall from the other end like this, है ना? Because the slide is slant. So basically the Leifson stain is also flowing all over through the inoculum. Right now what we are going to do is, again I'm going to wash the slide with the water so that any extra stain, if in case it's there, it will get washed off. And then I'm going to add methylene blue. Simple as that. The same thing, keep the slide slant position, add the methylene blue in this manner. Okay. Now once the steps are done, you will observe it through oil immersion through the microscope. Okay. You will use oil immersion method to observe it through the microscope. Now when you notice in the microscope, you will see that the flagella appears in red and bacterial cell appears in blue. Now just because we are using counter stain here, that is methylene blue, just because of which we can easily differentiate between the bacterial cell and the flagella, because flagella will appear in red in color and the bacterial cell will appear in blue color. So this is how it appears in Leifson method. Okay. So I hope the flagella staining method was very clear and understandable. I hope the video was helpful. All of you all the very best. I hope you give your best shot in your examination. Let's meet in the next part wherein I'm going to teach the third method, the semi-solid media method. Okay.